Hello guys, welcome back to another video. This will be our first biology video. In this video, we will be discussing how the structure of DNA determines the structure of proteins which help carry out the essential functions of life through specialized cells. I hope you enjoyed this video and let's get started. So what is DNA? DNA is short for deoxyribonucleic acid and is a molecule that's organized in the form of genes located in the nucleus of each cell. It's a double-stranded helix of nucleotides that contain the genetic code of the individual. So basically, a double-stranded helix looks like this, and it contains many nucleotides. Nucleotides are the basic units of DNA, which is made up of a phosphate, a sugar, and a base. There are 4 chemical bases for DNA, which consist of adenine, cytosine, guanine, and thymine. These bases pair specifically like A with T and C with G to form a double helix structure. When these bases are connected to a specific pair, it looks like the picture on the left. As we can see, adenine and thymine are connected, and cytosine and guanine are connected, and they are all connected and held together by the phosphate backbone. The next thing we need to know is RNA. RNA is very important because it relates to DNA by DNA acting as a permanent blueprint stored in the nucleus, while RNA copies, translates, and carries that information to build proteins. But what is RNA? RNA is short for ribonucleic acid and is a single-stranded polymer that is produced by DNA. There are 3 types of RNA, which are messenger RNA, transfer RNA, and ribosomal RNA. mRNA is a single-stranded molecule that carries genetic instructions from DNA in the cell's nucleus to the ribosomes and the cytoplasm. tRNA acts as an adapter, bringing specific amino acids to the ribosome. rRNA forms the ribosome structure, creating the machinery that reads the mRNA code to build proteins. This is crucial for protein synthesis. Protein synthesis is a 2-step process including transcription and translation that builds proteins from amino acids based on DNA genetic code. What is transcription and translation? Translation is the process of synthesizing proteins by decoding messenger RNA into a specific sequence of amino acids occurring on ribosomes in the cytoplasm. Transcription is where a specific segment of DNA is copied into a new single-stranded RNA molecule. Going back to protein synthesis, there's an easy way to memorize the steps. Just remember DNA to RNA to protein. First comes transcription, where DNA is copied into mRNA in the nucleus. Then comes translation, where mRNA travels to the ribosome and is read to create a chain of amino acids which form a protein. You can also think of it as copy, carry, create, where DNA is copied to RNA, RNA carries the message, and the ribosome creates the protein. One very important type of protein is enzymes. Enzymes are biological catalysts, which means they speed up chemical reactions in the body, such as digestion, respiration, and metabolism. Without enzymes, these reactions would happen too slowly to support life. The function of an enzyme depends on its specific shape, which is determined by the sequence of amino acids and ultimately by the DNA code. This means that the structure of DNA directly affects how enzymes function. Enzyme activity or reaction rate can be influenced by several factors. One major factor is temperature. As temperature increases, enzyme activity typically increases because molecules move faster and collide more often. But if the temperature gets too high, the enzyme can lose its shape and can no longer function properly. Another important factor is pH. Each enzyme has an optimal pH at which it works best. If the pH becomes too acidic or too basic, the enzyme structure can change, reducing its effectiveness or stopping the reaction completely. Substrate concentration also affects reaction rate. When more substrate molecules are available, the reaction rate increases because there are more opportunities for enzyme-substrate collisions. However, once all enzyme active sites are occupied, the reaction rate levels off. This connects to the lock and key model of the enzyme function. In this model, the enzyme is like a lock and the substrate is like the key. The substrate must fit perfectly into a specific region of the enzyme called the active site. Only the correct substrate can bind to the enzyme, which ensures that enzymes are highly specific in the reactions they catalyze. Overall, the structure of DNA determines the sequence of amino acids, which determines the shape of proteins including enzymes, and that shape determines how effectively they function in carrying out the essential processes of life. Thank you guys for watching and stay tuned for more biology videos. Bye!